Hello everybody, my name is Roman Bauer and today I'm going to talk about our research involving computational models of brain development. First of all, I would like to thank the organizers for having me. So we are interested in the brain, which is a highly complex organ, and the human brain has almost 100 billion neurons and 10 to the 15 synapses, which are the connections between neurons. So that's a 1 with 15 zeros behind it. And the brain is what we're studying here, but it's actually also a source of inspiration for other applications. And in this talk, I will present some of our research involving computation models of the brain, but also then brain-inspired principles that then can be leveraged for other applications, including AI. So one computational approach that we have been following in the past is so-called agent-based modeling. What is agent-based modeling? So in our case, in a biological system, we model how cells behave according to certain rules, which are genetically encoded, and also there are physical interactions in three-dimensional space. So, for instance, this could be mechanical interactions between cell bodies, or this could be chemicals that diffuse in three-dimensional space, or electrical activity, and so on. And it is this fundamental interaction between those two entities that then generates highly complex systems, biological systems, which we are modeling with computers because these are so complex systems. So those dynamics are extremely complicated because they are interacting with one another at different time scales and at different spatial scales. And so we need computer models to really to really make sense out of this kind of entangled processes. Now, importantly, we model the cell elements as autonomous agents. So this means that there is no orchestrator, global orchestrator that tells the cell elements what they should do. But as I said before, the agents or the cell elements, they are autonomous and they follow their own genetic rules. And of course, their external environment can also affect them, but there is not like a global supervisor that knows everything and that gives instructions based on its global knowledge. So that's an important part of any agent-based model, is this kind of self-organized, autonomous dynamics. Now, one example of a brain architecture that we have in the past studied with agent-based modeling is the so-called cortical layer architecture. So here you see a Nissl stain where you can clearly see different cell types arranged in this laminar fashion. So these are different cytoarchitectures for the different layers of the visual cortex in this case. And so this shows that for some reason, we don't completely understand why, but for some reason, there are these different cell types arranged in a layered fashion. And very interestingly, we see this across all mammalian species. And so here you see some examples from different mammalian species. And importantly, even though the fundamental layer architecture is the same, there are lots of variations of this layer architecture. So some of them are thicker, some of them have different cell densities than others, different cell numbers than others, and so on. And if you look at this quantitatively, so if you look at the number of neurons in different layers for different species, or even in the same species but in different parts of the brain, then you can appreciate that this varies significantly, and so not only proportionally, but also in terms of absolute numbers. And so we were intrigued by this because for some reason there is this versatility and flexibility of the layer architecture. And we wanted to see whether we could create a computational model that could reproduce this kind of versatility. So how did we do that? We created or we implemented an abstract gene regulatory network where we have abstract genes or genetic states interacting with one another. And we start from a single precursor stem cell state, S1. And this stem cell state can then differentiate into different neuron types. So these will be different neuron types. Marginal zone later on becomes layer 1, layer 6, layer 5, layer 4, layer 3, and layer 2. And we have also apoptosis. So A stands for apoptosis, which is cell death. And importantly, and I will get back to that afterwards, in actual brain development, mammalian brain development, more than 50% of all cells die naturally. So this is very astonishing, and it's very counterintuitive because for some reason evolution came to this kind of solution where a large fraction of all cells just die naturally, even in a healthy cortex. And our computational modeling here requires actually apoptosis in order to generate this kind of layer architectures that we have then also compared with experimental measurements. So here you see a sequence visualizing how these different layers develop in our simulation with agent-based modeling. You can see here that first the top layer, which is actually the marginal zone, develops, and then layer 5 cells, so here in cyan, they migrate through layer 6 and they push up the marginal zone. And then layer 4 cells push up the marginal zone by migrating through layer 6 and 5, and so on. So as you can appreciate here, the cortex actually develops in an inside-out manner. So it's not layer 1, layer 2, layer 3, layer 4, layer 5, 6, but it's actually that layer 5 develops via migration through layer 6, and then layer 4 develops through migration of layer 6 and 5. And so we have this inside-out development, which is the way that it happens in actual natural development. And that's how we also created our simulation with. And so we could reproduce this temporal sequence. And then we could also reproduce proportionally the right neuron numbers in the different layers. So we have compared that with experimental data. And here you can see a video playing this sequence out. So importantly here, this is actually a three-dimensional simulation, but we see it as a two-dimensional video because from the side we can appreciate all the layers. And so this is actually a three-dimensional simulation. And so there is a lot of cells here that interact with one another mechanically. So that's a very important part of an agent-based model, is that cells interact with one another physically. And this requires, of course, a lot of computational work, right? And so this is why agent-based models have the disadvantage often of being very demanding from a computational perspective. But in this case, we could generate some small but still realistic layer architecture based on biologically plausible rules. And also, we could actually reproduce certain characteristics of certain neurodevelopmental disorders. So, for instance, in polymicrogyria, the cortex is much thinner and has many more folds. So here on B, this is Here you can see histology from a polymicrogyria cortex, and this is a healthy cortex, so you can appreciate that this is much thinner than here, and we could actually reproduce a much thinner cortex also by just changing a single parameter in our model. And so we could potentially use our model to better understand how does polymicrogyria exactly develop, and then ultimately really correlate mechanistically during development and at what time exactly those important factors then are involved. And also we looked into autism spectrum disorder. So here in D, you see again visualization of cells stained, showing their quite irregular locations. So you see these clumps often here. They are not very regular. Here on the left-hand side, this is a control stain where you can see a much more regular location, much fewer clumping. Here, much more clumps, and we can actually reproduce similar characteristics in our model by just changing a single parameter again. And so this kind of developmental simulations can therefore help better understand how do those developmental diseases or disorders arise, and then potentially could even help with designing treatment strategies. So overall, this kind of approach allows us to gain insights into questions such as, for example, why is there so much cell death? Because in our model, we need so much cell death in order to reproduce this variation in cell architectures and layer architectures. So we suggest that cell death is actually important in order to allow the cortex to be flexible enough to meet evolutionary demands for different species and different brain areas. Because as I showed before, in different brain areas, you can have also very different layer architectures. And so we believe that cell death and apoptosis are crucial in order to allow or enable this kind of flexibility of the layer architecture. And that's also why the title of our paper is called Creative Destruction, because the destruction is kind of necessary in order to enable the creative aspect of architectural development. So these are explanatory, powerful models, but an issue is, as I said before, is the computational demand. And in particular in agent-based models, due to the many interactions, this is a serious problem. And so what we did in order to address this problem is we created an international consortium called Biodynamo that currently includes 8 organizations. And we have implemented a highly efficient software called the Biodynamo software, which is open source. So everyone can download it for free, can use it for free. Even commercially, you can use it for free, as long as you acknowledge the software properly. This is published under the Apache 2 license. And so it's something that can be used by anybody for free. And in this paper, we present applications of Biodynamo, and we of course present the way it is implemented. And so if you're interested, please have a look into this paper. And we have, of course, then continued our research involving neural network development or neural tissue development using agent-based modeling. Now, in this case, we can now really scale up our models very much due to the highly efficient implementation of Biodynamo. So we can really simulate highly realistic tissues on a large scale. And so it has been a very important milestone for our research in my lab to have this Biodynamo software. Now I would like to talk slightly on a different aspect, which is about learning from the brain in order to apply those principles, those learned principles, for applications such as, for instance, AI. And in this paper here, we present an approach for training deep learning models by mimicking some important aspects of the actual brain. So in this case, we have looked into topographical mapping and also into the way and the dynamics of how synapses develop and then afterwards are pruned. And our research was actually recently featured in BBC News, showing that this is something that is of interest to a large audience. So during natural biological brain development, there is initially an overproduction of synapses and neurons, and then afterwards a pruning stage where a lot of synapses die naturally, and then there's also specialization. And so if you kind of visualize this with a line, you have first a big overshoot kind of production of synapses, then a pruning, and then it only becomes less, unfortunately. With age, the number of synapses decreases, but there's a lot of specialization happening. And topographic maps are a very specific type of connectivity mapping. For instance, in the visual system, like in the retina, which is the neural tissue at the rear of the eye, we have topographic maps. There the photoreceptors connect in this kind of neighborhood preserving manner to the retinal ganglion cells. So we have neurons that connect to, that are near each other, will connect to neurons that are also near each other. So you have this very ordered way of connecting. And we have kind of copied or stolen this idea from nature. And we have created similar artificial neural network connectivities where the features, the input features, are topographically connected to the hidden layer and then to the rest of the network. And just to better clarify how exactly this is set up. So we have the input features, which can be, for example, pixels, and then those are connected to the first hidden layer, which is usually smaller than the input layer. So we have fewer neurons. And so what we do is we then repeat this kind of connectivity of neighborhood preservation and this mapping, these topographical maps in this manner. And so we end up with a very efficient connectivity scheme. So in contrast to traditional ways of initializing this connectivity, which is usually a dense connectivity, a dense input mapping, where all neurons here connect to all neurons here, we actually just use a topographical sparse mapping, which is much more efficient. And here you can see this play out. Initially, this visualizes the fact that we have here a lot of connections. And then after the pruning epoch, we end up with a much lower density of connections, right? Here we have a high density, here a low density, and then it further decreases a bit after specialization. At epoch 6, at the end of the training process, we end up with a sparse and highly specialized type of connectivity topology. So you can see how this over time evolves. And we have then also compared this approach, which we call ETSM, Enhanced Topographical Sparse Mapping. So here you see different data sets that we used as benchmarks. Here you see the density level after training with the different methods. ETSM, as I said, is our proposed approach that we present in this paper. And then you can see here that the bold fonts indicate the best accuracies. And usually this is the ETSM. method that performs extremely well compared to traditional pruning methods. And also, importantly, we have usually less epochs, so we need less time, training time to accomplish this training with even higher accuracy, which we think is also because there is less overfitting. And so we can here demonstrate that our proposed method actually performs very well and leads to very low density, very sparse connectivity. And so it's a more sustainable way of using AI because, as you know, AI is very power demanding. We believe that learning from the brain can actually make AI more sustainable. So overall, I've shown that agent-based modeling can serve as a powerful framework to capture biological or neural tissue dynamics. I've talked about our work on the BioDynamo software. I'm the spokesperson of that collaboration, and if you're interested in any applications of BioDynamo, potentially for your own research, please don't hesitate to get in touch. BioDynamo is very versatile, so it doesn't need to be on neural network development. It can be many other applications, like, for example, cancer growth or cancer treatment. We have also used BioDynamo for those applications, and also epidemiology. So the elements don't need to be cells, they can also be individuals or households. Even economic simulations can be done with BioDynamo. And we show that topographically organized connectivity has computational benefits. So we can take inspiration, as I said at the beginning, from the brain to improve AI. And also we can kind of take inspiration from the fact that there's a lot of pruning going on to make those artificial neural networks much more sparse, but still perform very well. So thank you very much for your attention. And if you should have any questions, please don't hesitate to let me know. Thank you.